Module 9 Behavior, Biology in the Mind. Your every idea, every mood, every urge is a biological happening. You love, laugh, and cry with your body. Without your body, your genes, your brain, and your appearance, you would indeed be nobody. Although we find it convenient to talk separately of biological and physiological influences on behavior, we need to remember to think, feel, or act without a body would be like running without legs. Our understanding of how the brain gives birth to the mind has come a long way. The ancient Greek philosopher Plato correctly located the mind in the spherical head, his idea of the perfect form. His student Aristotle believed that the mind was in the heart, which pumps warmth and vitality to the body. The heart remains our symbol for love, but science has long since overtaken philosophy on this issue. It's your brain, not your heart, that falls in love. In the early 1800s, German physician Franz Gall proposed that phrenology, studying the bumps on the skull, could reveal a person's mental abilities and characteristic traits. At one point, Britain had 29 phrenological societies, and phrenologists traveled North America giving skull readings. Using a false name, humorist Mark Twain put one famous phrenologist to the test. He found a cavity and startled me by saying that he had that cavity represented the total absence of the sense of humor. 3 months later, Twain sat for a second reading, this time identifying himself. Now the cavity was gone, and in its place was the loftiest bump of humor he had ever encountered in his entire long life experience. Although its initial popularity faded, phrenology succeeded in focusing attention on the localization of function, the idea that various brain regions have particular functions. You and I are living in a good time Gall could only dream about. By studying the links between biological activity and psychological events, biological psychologists are announcing discoveries about the interplay of our biology and behavior in a mind at exhilarating pace. Within little more than the past century, researchers seeking to understand the biology of the mind have discovered that the body is composed of cells. Among these nerve cells that connect electricity and talk to one another by sending chemical messages across a tiny gap that separates them. Specific brain systems serve specific functions, though not functions Gall supposed. We integrate information processed in these different brain systems to conduct our experience of sight and sound, meanings and memories, pain and passion. Our adaptive brain is wired by our experience. We have also realized that we are each a system composed of subsystems that are in turn composed of even smaller subsystems, tiny cells organized to form body organs. These organs form larger systems for digestion, circulation, and information processing, and those systems are part of an even larger system, the individual, who in turn is part of a family, culture, and community. Thus, we are biosocial systems to understand our behavior. We need to study how these biological, psychological, and social systems work and interact. In this unit, we must start small and build up from nerve cells to the brain, and then the environmental influences that interact with our biology. We will also work from top down. We consider how thinking and emotions influence our brain and our health. Neural communication. For scientists, it is a happy fact of nature that information systems of humans and other animals operate similarly, so similarly that you could not distinguish between small samples of brain tissues from a human and a monkey. The similarity allows researchers to study relatively simple animals such as squids and sea slugs. To discover how our nervous systems operate, it allows them to study other mammals' brains to understand the organizations of our own. Cars differ, but all have engines, accelerators, steering wheels, and brakes. An alien could study any one of them and grasp the operating principles. Likewise, animals differ, yet their nervous systems operate similarly. Though the human brain is more complex than a rat's, both follow the same principles. Neurons. Our body's neural information system is complexity built from simplicity. Its building blocks are neurons, or nerve cells. To fathom our thoughts and actions across memories and moods, we must first understand how neurons work and communicate. Neurons differ, but all variations are on the same theme. Each consists of a cell body and its branching fibers. The bushy dendrite fibers receive information and conduct it towards the cell body. From there, the cell's lengthy axon fiber passes the message through its terminal branches to other neurons or muscles or glands. Dendrites listen, axons speak. Unlike the short dendrites, axons may be very long, projecting several feet through the body. A neuron carrying orders to a leg, for example, has a cell body. An axon roughly on the scale of a basketball attached to a rope 4 miles long. Much as home electrical wire is insulated, some axons are encased in myelin sheaths, a layer of fatty tissue that insulates them and speeds their impulse. As myelin is laid down up to about age 25, neural efficiently judgment and self-control grow. If myelin sheath degenerates, multiple sclerosis results, communication to muscle slows, with eventual loss of muscle control. Neurons transmit messages when stimulated by signals for our senses or when triggered by chemical signs from neighboring neurons. In response, a neuron fires an impulse, called the action potential, a brief electrical charge that travels down its axon depending on the type of fiber. A neural impulse travels at a speed ranging from sluggishly 2 miles per hour to breakneck 180 miles per hour. But even this top speed is 3 million times slower than the electricity through a wire. We measure brain activity in milliseconds and computer activity in nanoseconds. Thus, unlikely the nearly instantaneous reaction of high-speed computer, your reaction to sudden event, such as a book slipping off your desk during class, may take a quarter second or more. Your brain is vastly more complex than a computer, but slower at executing simple responses. And if you are an elephant whose round-trip message travel from a yank on the tail to the brain and back to the tail is 100 times longer than for a tiny shrew, reflexes are slower yet. Like batteries, neurons generate electricity from chemical events. In neurons' chemistry to electricity process, ions are exchanged. The fluid outside the axon membrane has mostly positive charged ions. A resting axon fluid interior has mostly negative charged ions. This positive outside, negative inside state is called the resting potential. Like a tightly guarded facility, the axon surface is very selective about what it allows through the gates. We say the axon surface is selectively permeable. When a neuron fires, however, the security parameters change. The first section of the axon opens its gates rather like sewer cover flipping open, and positively charged sodium ions flood through the cell membrane. This depolarizes that axon section, causing another axon channel to open and then another, like a line of falling dominoes, each tripping the next. During a resting pause called the refractory period, rather like a web page pushing to refresh, the neuron pumps the positively charged sodium ions back outside. Then it can fire again. As in figure 9.2, the action potential speeds up by hopping from the end of the myelin sausage to the next. The mind boggles when imagining this electrochemical process repeating up to 100 or 1,000 times a second, but it's just the first of many astonishments. Each neuron is itself a miniature decision-making device performing complex calculations as it receives signals from hundreds, even thousands of other neurons. Most signals are excitatory, somewhat like pushing neurons' accelerator. Some are inhibitory, more like pushing its brakes. If acceleratory signals exceed inhibitory signals by a minimum intensity or threshold, the combined signal triggers an action potential. The action potential then travels down the axon, which branches into junctions with hundreds or thousands of other neurons, or with the body's muscles and glands. Increasing the level of stimulation above the threshold will not increase the neural impulse intensity. The neuron's reaction is an all or none response. Like guns, neurons either fire or they don't. How then do we detect the intensity of stimulus? How do we distinguish a gentle touch from a big hug? A strong stimulus can trigger more neurons to fire more often, but it does not affect the action's potential strength or speed. Squeezing a trigger harder won't make the bullet go faster. Neurons interweave so intricately that even with a microscope you would have trouble seeing where one neuron ends and one begins. Scientists once believed that the axon of one cell fused with the dendrites of another is an uninterpreted fabric. The British philologist, I can't say it anymore, Sir Charles Sherrington, noticed that neural impulses were taking an unexpectedly long time to travel a neural pathway, inferring that there must be a brief interruption as in the transmission. Sherrington called this meeting point between the neurons a synapse. We now know that the axon terminal of one neuron, in fact, is separate from receiving neuron by a synaptic gap, less than 1/1,000,000th of an inch wide. Spanish atomists San Antonio, Romano, and Cal marvel at these near unions of neurons, calling them protoplasmic kisses, like elegant ladies air kissing so as not to mess up their makeup. Dentries and accents don't quite touch. No poet Dane. How do neurons execute this protoplasmic kiss, sending information across the tiny synapse gap? The answer is one of the important scientific discoveries of our age. When an action potential reaches the knob-like terminals at axon's end, it triggers and releases of chemical messengers called neurotransmitters. Within 100,000th of a second, the neurotransmitters molecules cross the synaptic gap and bind to receptor sites on receiving neuron as precisely as a key fits a lock. For instance, the neurotransmitter unlocks tiny channels at the receiving site, and ions flow in, exciting or inhibiting the receiving neuron's readiness to fire. Then, in the process called reuptake, the sending neuron reabsorbs the excess neurotransmitter. How neurotransmitters influence us. In their quest to understand neural communication, researchers have discovered dozens of different neurotransmitters, and almost as many new questions are certain neurotransmitters found only in specific places. How do they affect our moods, memories, and mental abilities? Can we boost or diminish these effects through drugs or diet? Later modules explore neurotransmitter influences on hunger and thinking, depression, and euphoria, addictions, and therapy. For now, let's get a glimpse at how neurotransmitters influence our emotions and our emotions. A particular brain pathway may be used only one or two transmitters, and particular transmitters may affect specific behaviors and emotions. But neurotransmitter systems don't operate in isolation. They interact, and their effects vary with receptors. They stimulate acetylcholine, which is one of the best understood neurotransmitters, play a role in learning and memory. In addition, it is a messenger at every junction between motor neurons and skeletal muscles. When ACH is released into our muscle cells receptors, the muscle contracts. If ACL transmission is blocked, as happening during some kinds of anesthesia, the muscles cannot contract and we are paralyzed. Researchers made an exciting discovery about neurotransmitters when they attached a radioactive tracer to morphine, showing where it was taken up in the animal's brain. The morphine, an opiate drug that elevates mood and eases pains, bound to receptors in areas linked with mood and pain sensations. But why would the brain have these opiate receptors? Why would it have a chemical lock, unless it also has a natural key to open it? Researchers soon confirmed that the brain does indeed produce its own naturally occurring opioids. Our body releases several types of neurotransmitter molecules similar to morphine in response to pain and vigorous exercise. These endorphins help explain good feelings such as runner's high and pain-killing effects of acupuncture and the indifference to pain in some severely injured people. But once again, new knowledge leads to new questions. How drugs alter neurotransmission. If indeed the orphans lessen pain and boost mood, why not flood the brain with artificial opioids, thereby intensifying the brain's own feel-good chemistry? One problem is that when flooded with opioid drugs such as heroin and morphine, the brain may stop producing its own natural opioids. When the drug is withdrawn, the brain may be deprived of any form of opiate, causing intense discomfort. For suppressing the body's own neurotransmitters production, nature charges a price. Drugs and other chemicals affect the brain chemistry at synapse, often by either exciting or inhibiting neurons firing. Agonist molecules may be similar enough to a neurotransmitter to bind to its receptor and mimic its effects. Some opiate drugs are agonists and produce a temporary high by amplifying normal sensations of arousal or pleasure. Antagonists are also bind to receptors, but their effect is instead to block transmitting functioning bolton. A poison that can be formed in properly canned food causes paralysis by blocking ACH release. These antagonists are enough like natural neurotransmitters to occupy its receptor site and block its effect, as in Figure 9.6, but are not similar enough to stimulate the receptor. Hura, a poison some South Americans Indians have applied to hunt dart tips, occupies and blocks ACH receptor sites on muscles, producing paralysis. Oh my God! In such animals struck by darts.